Hello students, welcome to my YouTube channel and please like, share, subscribe to my channel. Today we are going to start with the second chapter of class 12th, which is sexual reproduction in flowering plants. So number 1 is the introduction. Now the very first, embryology. So what is embryology? It is the branch of science dealing with the study of events which lead to developments of embryo. It is called as embryology. And now we have some names of scientists and their contribution to the plant embryology. Number 1 is Theophrastus. He wrote about the pollination in date palm. Next is the Andrew. He firstly find stamen as male organ of flower. Rudolf Camerarius firstly discovered sexual reproduction in plant. This is important for a NEET point of view. Next, Colleruter recognized the significance of pollination in seed setting and role of insect in pollination. He also studied the pollen morphology. Next is the double fertilization was discovered by Navaschin in Fritillaria and Lilium in year 1898. Next is the Strasburger discovered true fertilization in Monotrapa plant. Next is the Professor P. Maheshwari, known as father of Indian plant embryology. The name of his book is An Introduction to Embryology of Angiosperms. Next is the term alternation of generation was first used in plants by Hofmeister. Next is the in sexual reproduction, formation and fusion of gametes takes place, which are useful in development of new organisms. Next is the flowers are special organ that perform sexual reproduction in angiosperm. And the definition of flower is flowers are modified, condensed, reproductive shoots. This is very important. Next is the flowers are objects of aesthetic, ornamental, social, religious and cultural values. Next is the process of development of flower, firstly studied in Arabidopsis thaliana, which is a small annual plant related to Brassicaceae family. Next is the anthesis is the period during which a flower is fully open and functional. Now the next heading is the structure of a typical flower. This is the picture of a flower you can see on your screen. Now in a flower, this is the pedicel and the upper swollen part is called thalamus, and on thalamus all four whorl arises like sepal, petal, pistil and stamens. Now the sporophytic of angiosperm has well developed root, stem and leaf. देखिए एंजियोस्पर्म इज द एडवांस प्लांट और इसमें रूट, स्टेम, लीफ का कंप्लीटली डेवलपमेंट हो जाता है और इनमें सेक्सुअल रिप्रोडक्शन के लिए फ्लावर्स का फॉर्मेशन होता है. एक फ्लावर के अंदर डिफरेंट 4 वर्ल्स पाए जाते हैं और वो थैलेमस पे मिलते हैं. नंबर 1 इज द कैलिक्स, नंबर 2 इज कोरोला, number 3 is Androecium and number 4 is Gynoecium. So these are the 4 whorl found on thalamus. Number 1 is called Calyx. It is the first whorl made up of many sepals. The sepals are green in color, having leafy texture. The function of calyx is it provides the protection to inner floral part during bud condition. Next is the corolla. It is the second whorl. It has many petals. Now function, it provides protection and help in pollination by attracting insect due to attractive colors. Next is the Androecium. It is the third whorl. It is male reproductive organ and is made up of one to many stamens. It means stamens are the single unit of Androecium, and it is the fertile part that produces pollen grain. Next, number 4 is the Gynoecium. It is fourth wall. It is the female reproductive organ. It has one to many carpels. Now, next note is the Calyx and Corolla are called accessory or non-essential wall, while Androecium and Gynoecium are called as essential wall. Next is the some important similar terminologies. Carpel is also called as Megasporophyll. स्टूडेंट्स आपको ये टर्म्स याद करने पड़ेंगे आपके नीट पॉइंट ऑफ व्यू से, क्योंकि आपके ये नीट के अंदर कोई भी टर्म आ सकता है, तो आपको ये कंप्लीटली लर्न होने चाहिए. Ovule is also called as Megasporangium. Embryo sac is called as female gametophyte. Egg is called as female gamete. Stamens are called as Microsporophyll. Anther is called as Microsporangium. Pollen grain is Microspore. And last one is the germinating pollen is the male gametophyte. So these are the important terminology, or we can say homology, which are useful for your NEET exams. Next is the stamens. So it is the male reproductive organ. So you can see a picture of stamens on your screen, that these are two anthers which are lobe-like structure, and in between the anther you can see the connective, and this is the long filament, and this is the cut section of the anther. Next is the stamen is the unit of Androecium. It is male reproductive organ or microsporophyll of flower. Stamen has three parts like anther lobe, connective and filaments. Anther lobe, it is broader and fertile portion of stamen. So you can see this is the anther part, which is the broader part, and this is the fertile part. Next is the connective. Two anther lobes are separated in an anterior region by a deep groove. Both lobes are attached by a sterile parenchymatous tissues known as connective. Next is the filament. It is long and slender part of stamen. It connects anther to thalamus. So these are the three parts of a stamen. Next is the structure of anther. It is bilobed, tetragonal, oblong, knob-like fertile part. Each anther lobe or theca contains two pollen chambers or microsporangia, or we can say pollen sac. A bilobed anther is tetrasporangiate, means one lobe contains two chambers. So two lobes contain four chambers. In family Malvaceae, anthers are monoecious or monolobed and bisporangiate. Next is the monosporangiate anther is found in Arceuthobium. Next is the multisporangiate condition is found in Acacia due to septation. All four microsporangia of anthers are found at four corners of anther. They run parallel to one another. Each pollen sac or microsporangium is multi-layered structure. Next is its cavity is filled with pollen grains. The outermost layer of microsporangia is called epidermis. The microsporangia develop hypodermally. In another, from strip of archisporial cells. This type of development of microsporangia is called as eusporangiate. Now there is a difference in between eusporangiate and leptosporangiate. Now in eusporangiate, large sporangium develops from many initial cells and produce many spore. Example like Xylotum siliginella. Leptosporangiate, small sporangium develops from single initial cell and produce small definite number of spores. So this is the basic difference, that eusporangiate develops from many initial cell while leptosporangiate develops from single initial cells. Next is the each archisporial cell divides periclinally to form outer primary parietal cells and inner primary sporogenous cell. So now what is the periclinal division? Now you can see on the screen that there are two types of division found in a cell. One is the periclinal, and other is the anticlinal. So in periclinal division occur parallel to surface. देखिए ये सेल है और सेल में जो डिवीजन पैरेलल टू सरफेस हो रहा है तो उसको बोलते हैं पेरिक्लाइनल, और ये सेल है इसमें डिवीजन परपेंडिकुलर टू सरफेस हो रहा है, इसको बोलते हैं हम एंटीक्लाइनल. सो डिवीजन अकर पैरेलल टू सरफेस कॉल्ड एज पेरिक्लाइनल. इफ डिवीजन अकर लॉन्गिट्यूडनल टू सरफेस कॉल्ड एज एंटीक्लाइनल. अब जब डिवीजन सरफेस के पैरेलल हो रहा है तो वन सेल जो है वो बनेगी आउटसाइड, सेकेंड सेल बनेगी इनसाइड. सो न्यू सेल्स फॉर्म आउटसाइड एंड इनसाइड. और अगर डिवीजन हो रहा है एंटीक्लाइनल तो वन सेल बनेगी लेफ्ट साइड, सेकेंड सेल बनेगी राइट साइड. So you can see the difference in between both of division on your screen. Next is the archisporial cells are responsible for the formation of anther lobes, and it divides periclinally. Means parallel to surface, means one cell forms outside and second cell forms inside. So the outside cell is called as primary parietal cell. Inside cells is called as primary sporogenous cell. And again due to the periclinal division, the primary parietal cells forms four wall of anther like epidermis, endothecium, middle layer and tapetum, while primary sporogenous cells forms pollen grain with the help of meiosis. Now you can see the picture on your screen. There are four wall like epidermis, endothecium, middle layer and tapetum, and inside microspore mother cells are found. Next is the epidermis. It is the outermost protective layer. It is made up of single layer of cell. The anticlinal divisions occur in this layer which increase the size of anther. These are greatly stretched and flattened in tangential direction. Next is the endothecium. It is found under the epidermis. It is made up of radially elongated cells. The cells of this layer have fibrous thickening of alpha cellulose on inner side of radial wall. Next, fibrous bands are U-shaped and presence of fibrous thickening, it is called as fibrous layer. Next is the fibrous bands make the endothecium hygroscopic. ये moisture absorb करने की capacity रखता है. So it is called as hygroscopic. This layer plays key role in descent of anther. Descent means फटना anther and consequent liberation of pollen grain. क्योंकि जब एंथर बर्स्ट होता है, एंथर फट जाता है तो पोलन ग्रेन एनवायरमेंट में रिलीज हो जाते हैं फॉर पॉलिनेशन. फाइब्रस थिकनिंग इन दिस लेयर आर नॉट प्रेजेंट बिलो स्टोमियम और लाइन ऑफ डिसेंस. देखिए वो लाइन जहां से एंथर फटता है, एंथर डिसेंस होता है, उसको हम स्टोमियम बोलते हैं या उसको हम लाइन ऑफ डिसेंस बोलते हैं. स्टोमियम पर जो होता है इस तरह का डिपोजिशन नहीं होता है. नाउ व्हाट इज स्टोमियम? इट इज द साइट ऑफ डिसेंस and is made up of bulliform cells or motor cells which are thin-walled modified epidermal cells. Now notice this layer is not found in family Hydrocaritaceae and Clistogamous flower. देखिए Hydrocaritaceae प्लांट जो है एक्वेटिक प्लांट होता है और क्लिस्टोगेमस फ्लावर वे फ्लावर हैं जो कभी ओपन नहीं होते हैं. उन प्लांट्स के फ्लावर के अंदर ये लेयर प्रेजेंट नहीं होती है. Next is the middle layer. It is found below the endothecium layer. It is short-lived and made up of 1 to 3 layers of thin-walled cells. It provides nutrition to developing pollen mother cells with the help of tapetum. Note, it is not found in plant family of Laminaceae and Najadaceae. Next is the tapetum, which is really important for the NEET exams. It is found below the middle layer. It is innermost layer of Anther. It surrounds the microsporogenous tissues. This layer is characterized by polyploidy or multinucleate cells due to endoploidy. The part of tapetum which is found at the periphery or outside is known as P tapetum, while inner part which is origin from connective tissues known as C tapetum. Next is the DNA content is enormously increased in tapetal cells due to मल्टीन्यूक्लिएट कंडीशन. देखिए वहां पर न्यूक्लियस का नंबर जो होता है मोर देन 1 होता है, इसलिए वहां पर डीएनए कंटेंट इंक्रीज हो जाता है. नंबर बी इज एंडोमाइटोसिस. सो व्हाट इज एंडोमाइटोसिस? क्रोमोसोम डुप्लीकेशन एंड सेपरेशन ऑफ क्रोमेटिड्स टेक्स प्लेस विदाउट स्पिंडल फॉर्मेशन. नंबर सी इज पॉलिटिनी. इंक्रीज in the number of chromonimeta per chromosome is called polyteny. Next is the restitution nuclei. Up to anaphase, normal mitosis occur, but two sets of chromosomes thus formed are present in a common nuclear membrane. Such nuclei are called as restitution nuclei. Next is the note. If tapetum gets destroyed, then sterile pollen grains are produced. Next is the tapetum is of two types, like amoeboid, invasive or periplasmodial tapetum. In amoeboid tapetum, in this type the inner radial walls are dissolved, so protoplasm of these cells reaches into anther cavity and pollen sac. Protoplast in pollen sac is known as periplasmodium, which provides nutrition to the pollen mother cells. These cells absorbed all stored food of cells of middle layer, so cells of middle layer get died. Example typha, helianthus, elisma and tradescantia. This is called as amoeboid. Number 2 is the secretory, glandular or parietal. In this type, the cells are always found at the fixed position. Its cells secrete food material for developing microspore. In this type, tapetum absorbs food material and provides regularly to the developing microspore, so middle layers survive for a longer time. Next is the functions of tapetum. It provides nourishment to the developing microspore mother cells and pollen grains. It secretes hormones like IAA, means indole acetic acid, and enzymes. It secretes enzyme like Calyx, responsible for degradation of callose wall around pollen tetrad. It produces special protein for pollen grains to recognize compatibility. देखिए compatibility means अपने ही pollen grain को पहचानना. Next is the it produces lipid rich ubis granule containing sporopollenin for exine formation of pollen grain. देखिए pollen grain पर 2 कवरिंग होती है, outer is exine, inner is intine. तो exine formation जो होता है वो sporopollenin से होता है, और sporopollenin में जो होता है वो ubis granule मिलते हैं जिनका फॉर्मेशन लिपिड से होता है. ये बहुत इंपॉर्टेंट है. Next is the it also produces pollen kit, which is oily and sticky covering of lipids and carotenoids found over the pollen grains in case of entomophilous flowers. Entomophilous flower means जहां पर pollination जो होता है वो insect से होता है. Next is the pollen kit also provides protection the pollen grains from UV rays. Next is the it provides specific smell which is useful to attract the insect. The cells of tapetum possess numerous spherical bodies called proubis bodies or proorbicules. They are lipid nature. At maturity of pollen grains, proubis body comes out of tapetum cells into the space of cell membrane, where they unite with sporopollenin to form ubis body or orbicules. Ubis body provides help in the formation of exine layer of pollen grain. Now you can see the developmental stages of anther. Number A diagram is the initial stage, and in B there is archisporial cells found in all four corners, and centrally procambial strand is formed. And now in archisporial cell, due to the periclinal division, देखिए it means parallel to surface, इस डिवीजन की हेल्प से यहां पर फोर वॉल का फॉर्मेशन हो रहा है और इस तरह से फर्दर एक कंप्लीट मैच्योर एंथर का फॉर्मेशन होता है. एंड वी कैन सी देयर इज फोर वॉल लाइक एपिडर्मिस, एंडोथीशियम, मिडल लेयर एंड टेपिटम. और सबसे इनर साइड में माइक्रोस्पोर मदर सेल मींस एमएमसी होती है, विच आर द डिप्लॉयड सेल्स. और ये इसका फर्दर आपको एनलार्ज व्यू मिलेगा, जिसमें सबसे आउटसाइड में एपिडर्मिस है, देन एंडोथीशियम है और 1 to 3 लेयर्स मिडल लेयर्स हैं. देन इनसाइड यू कैन सी द मल्टी न्यूक्लिएट स्ट्रक्चर ऑफ टैपिटम, एंड सबसे इनसाइड में पोलन सैक्स हैं और उसमें माइक्रोस्पोर मदर सेल्स जो हैं वो हेल्प करती है पोलन ग्रेन फॉर्मेशन में. नाउ like, share and subscribe, and thanks for watching. Have a nice day.